Welcome, and thank you for joining us for this U2U virtual lecture hosted by U alumni. I am Stephen Finkelstein, Advancement Director for Genomic Medicine here at the U, and it is my pleasure to introduce Dr. Nels Elde in today's lecture, The Viruses of Our Lives, Pandemics Past, Present, and Future. Dr. Elde is an Associate Professor of Human Genetics and holder of an H.A. and Edna Benning Presidential Endowed Chair at the University of Utah. His lab studies evolutionary genetics and the impact of infectious pathogens on their hosts, including viruses such as HIV, smallpox, and SARS-CoV-2. Dr. Elde also teaches critical thinking and research for first-year graduate students in the university's molecular biology program. He co-hosts the podcast This Week in Evolution and was named a 2020 MacArthur Foundation Fellow. We will hear first from Dr. Elde with a question and answer session to follow. Throughout the event, you can use YouTube's live chat feature to submit your questions. Dr. Elde, thank you very much for joining us today.
Thank you so much, Stephen. It's really fun to be here. Thank you to the Alumni Association, to all the alumni, the other members of our engaged community. Really a fun honor to give today's lecture. I anticipate this will be about a half hour, maybe a few minutes longer than that, and so we'll have plenty of time for questions. There's already some great questions that have been sent in ahead of time, but as you're listening to the lecture, reflecting on any questions you have, please put them into the live stream on YouTube, and those will get funneled to me at the end so that we can keep the conversation going. So ask me anything as we move forward here. Okay, I'm going to get my presentation shared and up and running. If this doesn't work somehow, maybe someone can tip me off to that, but hopefully now you're seeing my title slide, The Viruses of Our Lives, Pandemics, Past, Present, and Future. And of course, with SARS-2, I think on all of our minds, still very much with us today as we're moving forward into the summer here, hopefully at the beginning of a successful kind of new vaccine era, advancing that. Okay, but before I get to the details of SARS-2, I want to take a little bit of a broader view of the viruses of our lives. And so what you're looking at here are cartoons of virus particles. And of course, viruses are super tiny. So how do we make pictures of them? Or why do we think they look like this? This is based on really powerful microscope images of virus particles. And this collection illustrate a diversity of viruses, but ones that have one thing in common. They make us, our species, humans, they make us sick. And so some real familiar faces here. Of course, HIV, human immunodeficiency virus, next to SARS, next to measles, next to rabies, herpes virus. The reason we might think that these are the viruses of our lives is actually each of us participating in today's lecture are probably infected with at least one of these viruses, if not currently at one time in our lives. The herpes viruses in particular are super successful and sometimes really quiet hitchhikers. We don't even know that we're infected with them. So as Stephen mentioned, my lab is interested in the evolution of viruses. We focus on mainly this one in the upper left called Poliovirus. This is the causative agent of smallpox, a pandemic past, the only virus that we've completely eradicated using a very distributed vaccine strategy that was really successful. No smallpox in the US for decades and completely eradicated back in the 1970s from the world. So a real success story there. Obviously, other viruses that are more on our radar currently are ongoing pandemics, whether it's HIV or influenza that we might talk about as a past pandemic, but one that echoes on now as seasonal flu with various amounts of impact. And then some of the more scary viruses, Ebola, SARS, whether it's one or two, or perhaps pandemics of the future. And so we'll talk about that. But in my lab, we think about how these viruses evolve. And then on the other hand, how we as hosts of them sort of evolve or counter-evolve with them. And so this has led to a really useful concept illustrated here in this cartoon of the Queen of Hearts. And so this is borrowing from the author Lewis Carroll in his book, Through the Looking Glass, where the Red Queen, the character, says to Alice that it takes all the running you can do just to stay in the same place. And so as evolutionary geneticists, we've sort of adopted this idea to describe the mutational process whether it's a virus or a host species, with mutations accumulating, being selected either for temporary advantages, the virus might replicate better in a new host, or the host may have some variation that's selected on because it blocks a virus from being successful. And then this leads to these ongoing sort of genetic chases or even arms race sort of like scenarios that we think about to conceptualize our field. And so here today, we might put on the two sides of the arms race, SARS-2 and our own species, humans, obviously. And so as many of you are probably already thinking, there's a kind of a big issue here, which is these viruses evolve really rapidly. They accumulate mutations really quickly and can pose a tremendous challenge. Whereas as humans, our own species, our generation times are in the decades, and our mutation rates are relatively low. And so how is it that we're here at all? And so we think about that question. For today, I'm going to bend the rules a little bit. We're going to talk about virus evolution, the very rapid evolution that we're even observing today with SARS-2, thinking about some of the variants, so-called variants of interest, concern, and so forth. And then instead of thinking about our evolution, actually to think about how humans are intervening, whether it's with vaccines, natural immunity, our behavioral changes, our public health measures, and how that might be sort of driving the next wave of pandemic evolution here. Okay, so let's spend just a moment to sort of become a little more acquainted with SARS-2. So this is a coronavirus. Relatively for this class of viruses, this flavor of viruses, a very large genome. So 30 kb, this is kilobases, thousands of letters. So 30,000 nucleic acids. This is that four-letter alphabet, the A's, T's, C's, and G's of the genetic code. This is a DNA copy. I'm just showing the first 1,000 or so letters. So if I showed you the whole genome of SARS-2, it would run for 30 pages down the screen here. And I think it's really incredible, actually, to think about the fact that what something can do, what in this case, a genetic entity, a virus can do with only 30,000 letters in a four-letter alphabet. Imagine just sitting down at a keyboard and being able to type 30,000 letters that might have this kind of consequence. It's mind-boggling, actually. And so the fact that through the evolutionary process over thousands, even millions of years, this virus has emerged with this combination of letters for the first time that we've witnessed is really sort of tremendous testament to how powerful viruses are as replicative agents, agents of evolution, agents of replication. And so at the same time, we as humans have begun to learn a lot about how we can, for the very first time, really classify these things. And so in truth, sort of documenting those 30,000 letters was a snap, given a lot of genomic technology available to us. But understanding those 30,000 letters is much harder. And in fact, just comparing those 30,000 letters between isolates of the virus that we pull out of patients and then sequence their genomes sort of in real time over the course of a day or two, this has led to a tremendous, like a massive data set, millions now of whole SARS-2 examples of SARS-2 genomes taken from patients that we can analyze and try to squeeze some meaning from them. And so I thought I would just give one quick example of how having all of this information, this genomic scale information, can lead to perhaps useful ideas that might even tell us something about where this virus came from in the first place. Okay, so in this slide, this is another way of looking at the SARS-2 genome. So now instead of 30,000 letters, I'm showing you some bars. So working from left to right, from 0 kilobases or thousands of letters out to 30,000. And the cartoons here show domains. These are the regions of the genome that encode genes that get converted into proteins that allow the virus to conduct the business of replication as it infects a host cell. And so there are two small boxes that I sort of want to emphasize here. One is this RDRP, this alphabet soup stands for the RNA-dependent RNA polymerase. This is the enzyme that actually takes the one copy of the genome and makes another copy of that. So a core or key function for the virus to be able to replicate. The second gene encodes a protein called spike, which I think we've all heard of. This is the on the surface of the virus particle. the protein that sticks out, the spike protein, and addresses the host cell, finds the host receptor, binds with it in a way that facilitates the fusion of the virus particle to a new cell that's infecting, and also is displaying a shape that our immune system is very interested in. It becomes perked up as it recognizes a spike protein, responds to that by creating other proteins, antibodies, that alarm or set off the alarm of the immune system to try to block the replication of the virus with various immune defense strategies. And so given the fact that we had the sequences, I think when this research that I'm describing was done a few months ago, something like 200,000 of these 30,000 letters that you could just line up and compare. And so some evolutionary scientists looked and asked, what are the regions actually that look like they've been changing the most rapidly as you compare these viruses in recent time, even over sort of the course of a year? And so what they found, or what they proposed, are 7 regions of the genome. Some of them, most of them, are over spots that have those, that encode those genes that might be sort of changing the most rapidly. And the inference here is that that might give us a clue about where SARS-2 came from. Where did it spill over? So we presume from a bat species to humans, what were the genetic changes that might have been important for that virus to gain a foothold and to start replicating in our own species? And so what they did was to look at the original, the very first SARS-2 that was isolated in Washington state, and then ask if there are any differences in that virus compared to other coronaviruses related to it. And they've actually found one really interesting change. It's in the spike gene, the code for the spike gene, and there's a single letter of the alphabet is changed. So in SARS-CoV-2, there's a G that forms the part of this codon or 3-letter code to have an amino acid that is part of the protein of the spike, a single letter of protein. And that single change G is different from every other coronavirus that we've ever documented before, from bats, SARS-1. In all of these cases, you find an A letter in that position, whereas in SARS-2, it's a G. And in fact, now we're at about 1 million comparison points of SARS-2, and in every case, it's always a G there. And so that might be a clue about what was one of the key genetic changes that allows SARS-2 to replicate better, cause the symptoms of COVID-19. And in fact, the authors went on to actually reverse that change. So instead of having a G at that position in SARS-2, to engineer a virus that has an A there. And what they found is that it replicated a little bit worse in human cells relative to the ones that have the G. And so we can go very quickly from this like massive collection of genome scale data into the millions of samples and quickly pinpoint changes that we think might be important for why SARS-2 is a current or ongoing pandemic. Okay, so that's maybe a little hint at where SARS-2 came from. Now let's switch over in this Red Queen hypothesis to the human side and start thinking about the interventions that we've started to deploy in the last year or so since this virus emerged sometime around the turn of 2019 into 2020. And so, in addition to all of the public health related behavioral changes that we've made, whether it's social distancing, mask wearing, et cetera, hand washing, one of the really major pivotal advances of the last few months has been deploying the vaccine. And so in this graph, I think is going to maybe go down in history, certainly of our generation, as one of the biggest scientific breakthroughs. These are the messenger RNA-based vaccines that we've recently deployed. This data comes from clinical trial with the Pfizer vaccine, the BioNTech vaccine, and was reported in the New England Journal of Medicine about the start of 2021. And what we're looking at here on the y-axis, or going from bottom to top, is the cumulative incidence, or the percent of people who got either the placebo or the actual vaccine, the percent that have COVID-19, that are positive for the virus. And that's compared to the vaccine arm of the trial, the half or so of the people who enrolled in the trial who received the vaccine. And working from left to right, you can see the days after the first dose. And so if you look at the very bottom left part of the curve, you can see this overlap between orange line and blue line. And then as time goes on, post either vaccination or placebo treatment, you can see the difference here where the vaccine cases just fall off the map, and it's not even close. And so this is really a once in a generation, perhaps, breakthrough in our ability to very quickly turn the tide on this virus, given how efficacious, how efficient this vaccine has turned out to be, using some new technology to genetically program it to recognize or to deploy a part of that spike protein so that our immune system learns what that looks like and is sort of ready to go with a real exposure to the actual full virus itself. So I'm happy to report personally, I'm on the orange curve here, and at about the 21-day post dose 1 curve, and I can assure you in the audience and also my mother that I have an appointment to get a haircut and to sort of clean up the beard region here as well. That's going to happen in the next week or two. This sort of breakthrough has also just gotten me thinking, or almost daydreaming a little bit, about how we as humans deploy needles, and the idea that, you know, we can put ink into these needles and get something like a permanent tattoo. So here is a cartoon of a coronavirus. Perhaps that dagger is our progress even so far with the vaccine going forward. But I've been thinking about, you know, these vaccines almost like invisible ink in a sense, and that we're, you know, instead of visible permanent ink, this temporary mRNA dose, which actually causes our the cells to make that spike protein so that our immune system perks up to them. I think it's a really interesting sort of analogy here almost. This isn't myself, so it's not. This is from scraping the internet. I haven't made. I'm very comfortable with temporary invisible ink vaccine tattoos, less so with the permanent ink-based ones. So I've kind of been touting this vaccine, but what's some evidence that it's actually working, not just in clinical trials, but when we deploy it sort of in the wild, so to speak. And so here's already a somewhat dated from last month graph looking at SARS-2 cases in Israel. And there in Israel, so because of some agreements with their public health mechanisms and the Pfizer BioNTech crew, they've really moved quickly to get high numbers of people vaccinated. And so what we can see here is we're looking at now from bottom to top, the percent of the peak number of cases of COVID-19, percent of new critically ill, and daily death counts from COVID-19 in the country of Israel. And so just as, you know, the end of January and early February, as the vaccine really moved forward, first in older populations and then in the general adult public, you can see a crash in these percentages from peaks in terms of cases to critically ill and daily deaths. And so, of course, this is a more complicated scenario. It also involves you know, public health measures and other things like that, but it's pretty striking how quickly deploying the vaccine has really turned the corner on a lot of the cases, where now it's even hard to find new cases there to measure the numbers of virus spread going forward. So I wanted to pause, actually take a moment. So this is, you know, an ongoing pandemic, but let's look at some of the pandemics past and compare how our experience today might be different from pandemics that we have dealt with before. And so, skip the slide there. So this is another graph looking at poliovirus. And, you know, the reported cases of paralytic polio, this is shown in blue. So again, the case number is going from bottom to top, left to right is a time series year by year around the early 1950s. And so, you know, first some perspective. So in purple, we see deaths. And so the number of deaths per year attributed to polio, pretty modest, obviously, compared to COVID-19, compared to some of the seasonal flu years that we see. But pretty significant cases of paralysis. which then dropped precipitously over about a 8-year period with the introduction of the first polio vaccine pioneered by Jonas Salk. But this took years. And so the cases, we've really, I think in our current thinking, we've really contracted here as we think, almost by the weeks or months versus the number of years, to have some perspective on how long sort of deploying a vaccine and seeing the results can take. And so it wasn't until really 1963 when cases of paralysis were really disappearing, and not until even the late 70s when the US was declared polio free. Still not completely eradicated, some small pockets around the world, but really just on the cusp of polio potentially being the second eradicated pandemic. Okay, so now let's go back to today's pandemic SARS-2 and think a little bit about, as we've deployed the vaccine or continue to do so, and we have more supercharged immune responses, as well as a growing pool, unfortunately, of natural cases of immunity, in many cases success stories, but with sort of taking away the safety net and seeing the associated death toll as well. But so as the virus now faces basically humans that are armed with immune responses, either natural or vaccine mediated, what's happening with SARS-2 evolution? And so I think we're all pretty familiar with this notion of variants of concern. I would also say these might be considered tattoos, choices of concern as well. Different looks at how people are memorializing the current pandemic. But so let's talk a little bit about variants. What this means, how is the virus SARS-2 evolving kind of right before our eyes? And so here's another graph that tries to get at that, tries to illustrate the evolutionary process that we're seeing again, because we have sampled now millions of sequences, we can compare them and start to really learn about how the virus might be evolving. And so here in this graph on the y-axis, or working from bottom to top, we have the mutations counted across the genome. So how many letter differences or alterations in those 30,000 letters are there compared to a reference or one of the very first sequenced SARS-2 strains. And then again, time on the working from left to right over the months. And so what's emerging from these comparisons is that the virus makes between 2 or 3 mutations per month, about 23 a year across the whole genome. If you just sort of grab an average virus and ask how different is it after a certain amount of time. That's graphed mostly in those those blue dots. Those are each a virus sequence and how many mutations relative to the reference it encodes. And then the VOCs are variants of concern. And so I'll talk in more detail about this in the next few minutes. This really illustrates what's happened in the last few months. So we've had additional sets of mutations, a little bit off the graph, although not that much. And so the virus has sort of figured out mutational paths, it looks like. Has started acquiring a set of mutations that appears to allow the virus to replicate a little better, to transmit a little better, although the actual basis of that virologists are scrambling to figure out. But we've seen enough that it does look like the virus is adapting to humans as we've put up all kinds of roadblocks to try to block it, from vaccines to public health measures. So very busy kind of alphabet soup, but this is meant to illustrate 3 of the sort of household name variants of concern. So the first B.1.1.7, a lineage that first emerged from the United Kingdom, and a handful has about 17 or 18 mutations from reference, a handful of them that have been pointed out as potentially important. These are all mutations that map to that spike gene that encodes the spike protein on the surface of the virus. South African variant, as it's sometimes called, B.1.351, again, sort of alphabet soup here. And then P1, a lineage that was first seen in Brazil. I don't want to go into all the details here, except I want to point out those arrows. So those arrows between the variants are shared mutations. This is something called independent evolution or convergent evolution that evolutionary geneticists are very interested in, because it's basically the same mutation, almost like lightning striking twice or 3 times. You have these mutations that independently arose in different parts of the world. And that is a signal that this might be a clue of how the virus is adapting. In this case, for example, if we look at that N501Y, this is a mutation also because to try to get away from the alphabet soup, it's been called Nelly, based on, so those letters are the amino acids and the number is the position of the mutation in the spike protein. And so if we look at the mutation Nelly, we see that in all three variants. And so that sort of raises our interest that that's a mutation that matters for the virus replicating a little better. Perhaps it's evading some of our immune response. And there's some evidence that that's true, although some, thankfully, also evidence that are, for example, the vaccines are still quite effective against all three of these so-called variants of concern that have arisen very quickly in different geographies across the world. Here's another way of looking at these variants. So we're looking at the frequency, how often when you take the virus, you take a patient's sample from a patient, saliva or a related swab, and then sequence the genome of the virus and identify which variant is it. You can see the frequency here up to 1. And so gray doesn't indicate, actually, so there's always 100% viruses. The gray are not variants of concern, but the colored ones are indicating viruses that are in these lineages that are of interest or of concern. And so in red here is that B117, B.1.1.7 from the UK. And you can see as we moved into April, and that basically the only viruses that you recovered from patients were of that strain. It was transmitting better than any of the other viruses is the implication there. At the same time, overall numbers are way down in the UK. And so even as this virus has taken over, it's been harder and harder to be a successful SARS-2 virus in that country. Different story in the US. So here, the gray over much of 2020 is all kinds of different variants. Viruses mutate constantly, and so you have all of this variation, and none of them really had what looked like any kind of transmission advantage. It was sort of viruses competing, and that's even true today. So in red, again, that B117 sort of came up to about 50% frequency, but it sort of stalled out. And we've seen some other variants emerge out of New York, Michigan, some in California, but none of them have really sort of swept through the population the way that has happened in UK and other places in Europe. Again, very different stories if we look in South Africa. So the B1351 has dominated sort of the sampling that showed up there. And similarly in Brazil, this is that P.1 variant that came up in high frequency. Seems to have peaked out and is now hopefully coming back down. That's not the case everywhere. And of course, I think we've heard a lot in the news headlines recently about India. And so here's just a screen grab of daily new confirmed cases. And we've seen this massive spike sort of through the month of April, hopefully leveling off, but I think really indicating how combinations of the virus mutating, but then also I think what we're seeing here is really a healthcare system overwhelmed and what can happen when some of our basic sort of public health measures start to break down in a major way. Of course, people are really interested in, is there genetic variation here? Is there a variant that might be somehow involved in this? And the current answer is we don't know yet. There is evidence, and so here's that similar graph now showed in green is a different lineage called B.1.617. It's been around, actually, it's been noted for many months. So I think as early as last summer, and yet here we are into the spring 2021 or early winter in the Southern Hemisphere and seeing this outbreak, which correlates with this other variant, now variant of concern, although it's not clear, again, how much of this is sort of public health related, human behavioral, behaviors of concern versus a virus lineage of concern. And in the early returns on testing, for example, how the immune system responds to this different variant is looking favorable. This hasn't broken through the vaccines. And in fact, the vaccines have been really slow to be deployed in India. Natural immunity is relatively low as well, as they've been spared some of the major waves that we've seen, for example, in the US at the start of in January of 2021. Okay, so now I want to spend a few minutes thinking about future pandemics and bring in a little bit of work from my lab. And so this is a different flavor of coronaviruses, the so-called SADS related or SADS coronaviruses. And that is an acronym short for severe acute diarrheal syndrome. This is a coronavirus that has arisen recently in livestock and pigs in particular. In 2019, kind of under the radar, but entire farms where the pigs either were wiped out by the virus or had to be killed to try to slow the spread of the virus. And so this illustrates another point about coronaviruses in general, is that they move between hosts and spill over to new ones. And so we know something about that for SARS-1, where related viruses, very closely related viruses, have been isolated from bats, from civet cats, and then proposed that it was through the civet cats that they spilled over to humans back in the early 2000s. MERS, Middle Eastern Respiratory Syndrome, another coronavirus from about 2010, bats to camels to humans. And then SADS is one my lab has done some research and we would propose should be under some surveillance, that we should put this on the radar as a potential future pandemic, moving from bats to pigs. And we think potentially with the potential for farm workers or other people in contact in sort of commercial agriculture scenarios where we could see potentially a spillover of the future. So the work in a few slides that I'll show in about 5 minutes before we get to questions is done by Stephen Goldstein, a postdoc in my lab, who got his PhD studying the virology of coronaviruses, the virus biology of them. Stephen has really taken advantage of the last year to grow his experience into thinking about virus evolution, coronavirus evolution. You can hear a really interesting interview with Stephen from a couple months ago on Science Friday with Ira Flato on NPR. Will our vaccines work against the variants of concern, variants of the coronavirus? Again, the early returns are actually very positive, and Stephen goes into some details about why we should be feeling optimistic as we move into hopefully a vast deployment of the vaccines. The work I'll be telling you about was also showed up. Stephen was describing some of the work of how coronaviruses actually change their genomes, not just letter by letter, but in some cases chunk by chunk, masters of mixing their genome, and how we think this is an important thing to consider perhaps with pandemics of the future. Okay, so this is kind of complicated slide, shows how we can compare not just SARS-2, but in this case, coronaviruses, but this other class, the SADS related or SADS-R coronaviruses. And so we're looking at the genome again, not letter by letter, but by sort of left to right here. And each of those horizontal lines actually represents a letter, just as a different color. So there's 30,000 colored ticks there, all kind of scrunched together. And then below, we're comparing it to other SADS-related coronaviruses that have been isolated and sequenced over the last decade or so. And each of those slashes indicates where there's a genetic change from the top virus, the SADS-R coronavirus. Here's another way of just sort of quantifying how the changes sort of stack up based on the position in the genome. And so when you see the lines sort of separate from each other, those are, and especially those sort of valleys that almost appear as we compare some of the genomes to each other, color-coded here. These are indicative of entire swaps of sequence, where it's not just a letter change, but the virus, that polymerase, that RDRP, that copies one genome to make it two. In the case of co-infections, we'll jump to a different genome and start replicating that one. And you end up then with these recombined, this process of recombination, where the virus is almost a hybrid or a chimera of two viruses. And the fact that we see these valleys indicates that these viruses are actually swapping genetic material with very different coronaviruses pretty frequently. And so Stephen went in and mapped or proposed these so-called recombination regions, RRs, and numbered them for each of the viruses. And so you can see that pink virus, which is a coronavirus from bats, that's in this very closely related to the SADS virus, appears to have recombined 5 times with other viruses some of which we've never actually seen before. We don't have a copy of them in our databases. And so if we zoom in on this spike region, in particular, we can see for 1, 2, 3, 4, 5 of the, or 6 of the 8 viruses that we sampled, we could find recombination regions there. And so if we put this onto a sort of a tree of phylogenetic tree or a tree of viruses comparisons, each of those triangles or those arrows shows a recombination event where that virus was going along copying and it shifted onto a different virus and copied the other one to make a hybrid virus. Each of those large arrows are from viruses that we don't have a record of them in our databases. So it's retrieved or it's sampled a virus out in the environment that we haven't seen before. And so what this is telling us is that these coronaviruses are swapping a lot of genetic information, and that this could have implications for, this is a random process. Most of it is probably not of concern at all, but in rare cases, they might come to a combination where this is something where the virus that before recognized pig cells might recognize human cells, for instance, and that could be something to think about for a pandemic of the future as we think about these collisions with viruses, increasing collisions with them in the future. Okay, so I'm going to just wrap up here. I just wanted to mention this really fun, Stephen mentioned this at the beginning, this really fun honor, which probably brings me here today to be named a 2020 MacArthur Fellow. You can check out, there's a short video if you're interested in hearing a little bit more about our work and why it was recognized. If you go to the MacArthur website, you can see a short interview describing some of our work, not just with SARS or thinking about the current pandemic, but other cases of virus evolution, immune system evolution that we're working on here. I think this is just, you can go over to the MacArthur YouTube channel. I'm only, 400,000 views behind Lin-Manuel Miranda, and they just reopened Hamilton. So I need as many views as possible. So if you have a chance, maybe check that out. I want to just quickly thank the team here in my lab. So and some of our funders have supported this. And then in particular, Aaron Quinlan is another professor here in human genetics at Utah, and staff scientist Joe Brown, who helped us build that analysis pipeline that I was just showing you from Stephen's work on perhaps future pandemics and sort of new tools for us to gain better surveillance of coronaviruses, but other viruses that could be pandemics of the future. So I'm going to stop there. I think I'll end my slideshow, and then we can start to field some questions as they come in. I think Stephen's going to come back and read a few of these.
Well, first of all, Dr. Elde, thank you so much for that. That was fantastic. I'm sure the audience, as much as I appreciate the perspective that you brought, and we are absolutely privileged to have you working with us here at the U. So let's just go ahead and dive into some of the pre-submitted questions. And then if you have any questions out there in the audience, please feel free to send that on in with the YouTube chat that is live and active, and I will receive those questions as well. To start us off though, let's talk about the most previous once in a lifetime, once 100-year pandemic, 1919. Can you compare that pandemic to what we're seeing today?
Yeah, great question. And so this is the 1918 influenza pandemic, also called, that stretched into 1919 and beyond. Really fascinating. So two very different viruses, influenza versus coronaviruses. So the good news here is that these viruses don't themselves swap genetic information between them. There's no sort of super influenza coronavirus. They're different in many ways, but I think really an important comparison point. So first of all, obviously, here we are 100 years on, very different technology. Our knowledge of viruses, not just sort of the virology, but also sort of our ability at the genomic scale to measure these things, whole different ball game. And so very life-changing impacts. Some of the things that are not only the things that are different, but the things that are the same. So, realizing that influenza is a respiratory virus and deploying masks and social distancing. This is sort of 100 years old technology that can actually be pivotal, crucial in some cases still today. Obviously, I think our medical interventions, certainly at the hospital, that support level, very different. But as we know, seasonal influenza, not the exact same flavor as the 1918 influenza, but that we incur pretty serious health consequences year in and year out. We've become sort of familiar with the flu, and so almost don't blink an eye, I would say, with sort of relatively heavy mortality rates from year to year. Also, I think some really interesting comparisons on the vaccine, right? So influenza has been notoriously hard to get an effective vaccine from year to year. This is so these viruses do a different flavor of recombination called reassortment, where they take in, it's not a single piece of RNA, it's 8 segments or 8 separate pieces of RNA that as the virus is infecting, they can just swap them in and out. It's sort of plug and play almost compared to the coronavirus. And so that has been a massive challenge. The coronavirus vaccine has proved, the mRNA vaccine has proved really incredibly successful, to the point where I don't think we're looking at yearly, inoculations, maybe a booster 2 years, 3 years from now. I mean, this is all speculation, but a very different virus in that sense. In terms of the total numbers of, fatalities, both, I think, really, global pandemics that sort of really change the conversation, change our lives in really fundamental ways. And so worth it to consider that history, how we are repeating it in some ways and doing things differently in others.
Absolutely. And going off of that, trying to figure out where we're headed or the future of it all, now that we can look at the history, we have a question here about mathematical modeling.
Yeah.
And so do we have a mathematical model? Do we know when we're working on one in which we can identify how SARS-CoV-2 produces variants? And the question goes on to ask, in other words, does a variant survive and propagate at a predictable ratio to the number of persons infected?
Yeah, great question. And so I think hopefully my slide kind of midway through the presentation got at this a little bit. So what's really unprecedented here is, and you can get this, you can go, so nextstrain.org, some of the other databases that actually take all of these samples, put them together, build trees. You can see how the mutations are accumulating over time. Some of those graphs that I showed, that's how we estimated that, 22 or so mutations per genome on average per year. And so that's not really a mathematical model, but it's a prediction from all of those observations of what we expect, sort of that regression line. And then we can compare that to what we're seeing in real time. I'd say the difficulty with just sort of a strict mathematical model here is that it's sort of, there are a lot of moving parts, a lot of variables here. And so as our immune systems have become perked up either naturally or through vaccines, supercharged through vaccines, that changes sort of the selective pressure. As I think I mentioned, it's harder to be a SARS-2 coronavirus today than it was a year ago. And so the mutation rate will stay relatively constant. But the number of people the virus is infecting day to day will change. And so the number of active infections will influence how many mutations sort of will arise each day. You can start to plug this in and try to make some predictions, but it's sort of a moving target in some ways. And so I think, sort of taking that empirical data, and the numbers are so massive that we can actually learn a lot in sort of in our surveillance efforts moving forward of what we expect in terms of the kind of character of mutations going forward as well.
And that touches upon this question as well. And you talked a little bit about it as far as comparing the 1918 to the current one. But as far as just once in a century pandemics, plagues, however you want to put it, are concerned, audience question about what do they have in common, I guess, more from like a macro standpoint? Like how have they all found their way to impact our existence in such a manner?
Yeah, no, so this is a great question. Kind of, by looking backwards, can we forecast in the future? Will it be another 100 years before we have sort of a pandemic of this big of global impact? I would say, it's worth it to even make more comparisons. So if we just think about coronaviruses in the last 20 years, so SARS-1 was at about 2000 or so, and MERS in 2010, and then of course COVID-19 near 2020. And but the difference is here, right? So SARS-1, I think there were something like 800 deaths or so, maybe 100 deaths or so with MERS. And the different features of the viruses matter. So SARS-1 actually very deadly virus, but that kind of gave it away. And also, so of course we're all very familiar these days with the notion of asymptomatic spread of SARS-2. This is a sort of especially, I think, insidious situation because most of us infected with it don't know that. And then that's coupled with this idea of super spreader. So some of us are more likely to actually spread it to others than others for reasons we don't understand, by the way. We're starting to try to get some traction on that question, some really interesting recent work from some virologists thinking about that. But so it's sort of these sort of set of key variables that I think will influence how big one of these pandemics goes. And so, SARS-1 really deadly and not asymptomatic. And so we could very quickly quarantine or effectively isolate cases. MERS-2, something similar to that. I think we see a similar thing with Ebola. It's the symptoms are so obvious that we can immediately sort of go to this, kind of quarantined or lockdown state, where with SARS-2, it's if you don't know you have it, you're gonna sort of behave as usual, and so that's caused a lot of problems. And so I think maybe that's still a rough thumb rule, is that once in a century, the sort of a virus will figure out all of those things just through trial and error, where that it could have this sort of global, really massive kind of pivotal impact on our species. People are speculating about that, right? So so there's certainly some things changing about how many humans are on the earth, how often are we colliding, things like air travel, our collisions with wildlife as we continue to build and expand our own footprint as a species. That could certainly, I think, bend the curve so that 100 years could become 90 years, could become 80 years as we go forward.
And just to remind the audience listening, please submit some questions if you'd like. More than happy to ask them. And going off of that, pivoting through to the impact of not only us as a species, but in our cultural experience. There's a lot of apocalyptic connections to what's happening now. Massive changes to how we run our daily lives and what our expectations for the future is or are. And so it's on the minds of some of our listeners of the connection to more of our cultural representations of apocalyptic events such as this. The one specifically given was Revelations in the New Testament. And from your standpoint as a scientist and as a collective member of our entire experience here, how do you see those kinds of connections reflected in our experience now? And do you get a sense that in the future, we might see some sort of similar impact on our expression of our reality after we've experienced this together?
Yeah, this is a really great question. And, Stephen, in my MacArthur video, I think I opened by saying, I come from a family of artists, scientists, and ministers. And so I think it really is important that we have these conversations, not just actually on the scientific level, but as you're saying, or as the question is raising, culturally or theologically or philosophically, how do we step through this not only kind of medically, but make sense of it as a species, make sense of it kind of in the context of history and how we kind of conceive of ourselves. And so, whether it's the book of Revelations or other traditions, I think there's a theme that emerges again and again, which is that humans we, our species and our stories, we get out of balance from time to time in some pretty major ways. And I'm thinking here about the crashes of civilizations in the past, things like that, where and maybe this is reflected in some of the poetry, some of the writing in the Bible, for instance. When we think about the plagues, I don't think we don't have locusts in our backyard right now, but I think if you go to the East Coast, you'll find all kinds of cicada bugs.
I was going to say, it broods right out now.
That's right. And so these sort of really almost tipping points or really big kind of out of balance scenarios. And I think there's massive lessons in reflecting on that, thinking about how we, not only as individuals, but as communities, as collections of people, how do we respond? Can we do better with nothing to do with science at all? Of course, that's my work that undergirds sort of my day job, but I think there's so much more we can do by having conversations about how we try to restore some balance. How do we value that? How have our ancestors valued that? And how do we share that, in different traditions? I think we've, that's underappreciated right now as part of the interventions that are at our fingertips, that we have the possibility to do better when we sort of draw from some of those traditions, some of those lessons.
And globally, are there any lessons that we've learned so that in the future we can have more of a construct so that we can see these pandemics coming ahead of time? Do you have any suggestions on how we might be able to respond to that as a community?
Yeah, so I'd say two things. So one, and what we're seeing, what we saw in our country, kind of early 2021, and what we're seeing right now in India, really illustrates how this isn't sort of a country by country problem. It's easy to break it down into those categories to try to document things as they unfold, but that this is we're kind of in this together. This is a global thing, and the course of it will necessarily depend on what happens, not just within our own borders, but around the world. And that applies for the future too. So how are we potentially collaborating or in sort of growing conflicts over the types of information we share, the amount of sort of cooperation or coordination in terms of, in particular, if we want to narrow in a little more scientifically, what does virus surveillance look like through the next decade? So we have all these incredible tools that I would say scientists have really pioneered and engineered, brought to the forefront, and we've seen that. And the fact that any of us on the live stream can go to a website and have at our fingertips hundreds of thousands of these sequences. How do we think about that, not just for kind of reacting to the current pandemic, but forecasting or surveillance into ones potentially of the future? Again, lots of possibilities there.
There's an interview I saw not too long ago with a virus hunter from about a year or two prior to this pandemic. And they pretty much mapped out a respiratory illness that they thought that would jump on over. So what are the chances you think we'll experience another global pandemic such as this within our lifetimes, let alone 100 years?
Yeah, so I think, that's where perhaps our kind of more recent history with corona. So as we're thinking about coronaviruses, so the seasonal flu actually was really fascinating. So this past year, there was basically no flu season. Probably has a lot to do with some of the interventions that we did social distancing wise, the masking, the 6 feet distance, et cetera. But looking at coronaviruses and that tempo of SARS-1, MERS, I was proposing this SADS virus as one to have on the radar. I wouldn't be surprised if, once a decade, something like this spills over into humans, something in that 5 to 10 year window. We'll still be dealing with SARS-2 for the foreseeable future. Again, that global response, I think, will dictate a lot of the timing of that and the sort of future interventions, whether those are boosters to the vaccines or other kind of things like that. But then it gets back, to that idea of when the virus is sort of the frequency or the probability of the virus having the features, the respiratory spread, the asymptomatic hosts who are also super spreaders, that will really dictate sort of the depth of it. And maybe, Stephen, this is kind of going a little bit off the exact question, but I think it's worth it to sort of step back and take an evolutionary view. So it turns out that these viruses, whether they're in bats or pigs or other species, they are replicating every day like crazy, like by the billions, by the trillions of virus particles. Most of it, luckily, most of virus evolution is just junk. They just make mistakes and they sort of fall away. But every day, humans are being challenged. The viruses are sort of sampling humans or challenging humans in these exposures. Most of it completely invisible and most of it totally harmless. And it's only those really rare ones where where the virus replicates well and then has those features where it comes together. But I think we really underestimate how our interactions with wildlife, how this sort of unfolds given different sort of cultural, features and styles, really sort of sets the table potentially for these viruses to have a chance to do all these like almost natural experiments with us every day. And so it's really amazing, actually, not sort of the flip of the coin, not how often these things break through into pandemics, but how often they don't. I mean, it's just the numbers are breathtaking for how many of these viruses sort of are sampling humans as potential replication factories.
Is climate change driving that ability for them to have replication factories in us?
Yeah, so I think, probably that's a growing and interesting area of research. So I think, the things that take something a little bit out of balance again, to kind of draw back on that example, become opportunities. So, evolution often works kind of in these sort of various tempos where there might be an adaptive step to one environment, and then once that adaptive step is reached, whether it's a population of animals or microbes, just sort of sits for a while. I think when something like climate change changes the environment, that certainly disrupts sort of any of that sort of quiescent evolutionary space. At the same time, I think it's more the indirect effects are probably the real game changers here. So as the climate changes, what are the things that are leading to that? There's more people, so there's more there's more air travel, for example. So there's more a global mix of people, and I think that's really some of the indirect effects that are probably most directly impacting pandemics now and pandemics into the future.
I have one more prepared question sent in. So anyone out there in the audience, if you would like to ask a question, now is the time. And we mentioned SADS, you have that on your radar. We talked a little bit about how certain pathogens give up their game pretty easily, depending on what they're doing. Are there any real red flags right now, some emerging pathogens that's not just on our radar, but provide an imminent threat to a similar pandemic occurring?
Yeah, so I'm going to kind of, so that is a little bit of forecasting. I'm going to punt on that and actually say that the current things that concern me are the ones that kind of the familiar faces here. So I'm thinking about influenza, and I don't think I doubt that we're in position to see another kind of 1918 level event anytime in the near future. If anything, sort of the very near-term trends are favorable, but because that virus is still reassorting, because our vaccines, it's sort of a guessing game. And hopefully, actually, some of the lessons of the SARS-2 vaccine might be adopted or moved over to future responses to influenza. But that's still a very major player as a seasonal in terms of impact on public health. That's a major one. HIV, the ongoing pandemic there. So certainly some massive strides forward in terms of interventions, triple cocktail therapy, pharmaceutically, or even sort of preemptive treatments with it. But we're still talking about infections into the millions, life-changing, potentially sort of outcomes here. We've, the pandemics we're familiar with somehow, don't sort of, have that same impact as something that's totally new or kind of comes as a surprise. Less worried, I would say, about Ebola, just because as we're discussing some of the features that sort of give it away, but more concerned about things like measles. So as we start making decisions about whether we're, vaccine hesitant or how we deploy vaccines going forward, some of the, I think, lessons of the past have, up until recently, in some cases we've started to let down our guard a little bit. And so in not too far behind that, we've seen cases where, because it's very rare to eradicate these things, that they can kind of come back and once a foothold is gained and you reach these, levels where the mutations can really accumulate and test, human immune systems in real time, there's reason for concern there. And so, kind of keeping our guard up for the viruses of our lives that we already know would be my prescription for moving forward as well.
Well, thank you very much for all of your work that you're doing in helping to keep us safe and educated and preparing ourselves for the future. Really cannot be understated how privileged we are and how wonderful it is to be able to have an opportunity like this U2U lecture series to share such wonderful work with the community here. With that, that seems to be a wonderful way to wrap up this presentation. In the future, we hope that we can host you for talks that are a little less of the macabre, since we're still in it right now. But all the same, thank you again for volunteering this time and talking with us today.
My pleasure. Good to be together as a community. And thanks again to the Alumni Association. Thanks to everyone tuning in. Thanks to you, Stephen, as well.
No problem. Thank you all. Have a wonderful rest of your day and stay healthy and well.